Hi, I'm Todd O'Hara. I'm currently at College Station, Texas A&M University, visiting for the first time Tarleton University. Thank you. It's definitely a pleasure to be here. I wanted to be here a long time ago. Finally, we worked out the logistics. I thank the Department of Animal Science for inviting me and the support to come here. It all started with my former dean, Dr. Eleanor Green, and her relationships with people here related to horses. But my interests go well beyond horses, so as you'll see. So yes, we're going to try and combine wildlife management, animal science, human health, public health into this seminar. If you've ever been to Alaska or are familiar with Alaska, it's really easy to do up there. So on the right is a moose, and on the left is a male stellar sea lion. I suddenly realized, I mentioned to Trinette that I seem to have a male bias on my title slide, but when we get into this, the males don't matter anymore. It's about the females and the pups and the females and the calves, okay? So just being honest about reproductive biology and conservation, who really helps with the recruitment when it comes to these species. So I'm gonna have to go out here. So we're gonna define what trace elements are. We've been talking a lot today about how we assess them, whether they're deficient, too low, or toxic and too high. There's a lot of biogeochemistry interests. I'll give some examples of that, but that's more of how the soil, the ocean, and so forth interact with biology to make these elements available to our biota. Then One Health, I'll be tying in the human subjects. I'm a researcher, so I have to call them human subjects, sorry, into this. Then we'll get into some examples of Aleutian Islands, which I'll show you where they are if you don't. That's where we've done some stellar sea lions research. We're also doing fish research. We've been funded by commercial fishing industry, NOAA, and other agencies to do the fish work, but I'm going to focus on the sea lions. And then other Alaska large animals, Alces, this is the moose. We'll go through them as an example of large animals that would be similar to maybe the large animals that you're interested in. What are trace elements? Well, the ones we call essential are required for homeostasis. So they have a known role in some process to maintain homeostasis or the health of the animal. Some are known to be inadequate, so that would be a deficiency, or some can be overabundant in some regions. This is well known across North America, and we've been talking about that all day, that some producers make big mistakes because they think they're in a mineral deficient area, but they're not, and they give supplements, and they develop their own problem. So this is something that we're interested in talking about in Texas. This occurs in Alaska as well. Adverse effects. Now with essential elements, it's deficiency and toxicosis. You can have too much of a good thing. Undergraduates in college probably know that. Or they're going to learn to. For non-essential, well, there is no real known role in homeostasis. For some of these elements, there's still research going on to determine, well, maybe there are some roles in homeostasis or health, but they're not real obvious. But in this scenario, we're mostly concerned about adverse effects, that is toxicosis, too much. Some people like to say heavy metals. In the context that we use, it's best not to use those. For one, it gets confused with music, and two, it only describes them as how they present in the periodic table. It has nothing to do with health, okay? I suffer from heavy metal toxicosis because my first album was Aerosmith. By the way, they're my band, not your generation's band. So assessing them, deficiency to toxicosis, this is primarily why I'm here, to see how we can improve this with our, I'm sorry to have my back to you.
Oh, it's okay.
I know, but that's not polite. I'm sorry. So I'm trying to, sorry? You're used to being ignored?
No.
So the idea is we want to base this on, I'm sorry, this is just how I am. It's my own psychoses. So we're trying to figure out, can we base deficiency to toxicosis on actual health status, not statistics, okay? So when we're talking about normal, we don't want it to be a biostatistical assessment, like 95% confidence interval. What happens if all the animals you studied were diseased? Well, you just described normal for what? A disease state. So we need to improve this. And so this means we need to think about acute assessment and chronic assessment. Acute would be like a blood sample. What's the status of the animal with respect to its blood sample? Chronic might be a hair or hoof sample, something that's integrated their status over time. You also need clear objectives of what it is you're trying to study, because then that helps decide whether or not, oh, we're dead. Oh, well, diet, water, blood, or other tissues. So this is really important in wildlife, livestock, and so on. Marginal. When it comes to marginal, we don't mean drop dead deficient. It could be subclinical, where the animal's less resilient. For us in the wildlife world, this means poor reproductive performance, poor recruitment. In other words, the cow looks fine, but she can't take a calf all the way through to recruitment. So if you assess her health, it looks like everything's fine, but she can't support a calf. In one drainage I worked in for 3 years, we had 0 calf recruitment of moose. That became a problem. Marginal deficient, again, less resilient, to outright disease condition where it's obvious the element deficiencies led to a disease process. So we're interested in all this. High or toxic, it can be a stressor to outright toxicosis. So this is important to recognize. This occurs for essential elements too. So again, we want to develop our reference ranges for elements based on health status. And this is where we need the veterinarians and the animal scientists working together. If it's the veterinarian and the wildlife biologist working together. This relates to condition assessment. Hormesis. This is a really important concept for those of us that work in this business. This.
Can we take pictures?
Sorry. Autographs are after the show. Beneficial dose is, think about this as an essential element. If it's deficient, it's not so beneficial. But as you get to your optimal level, you've reached the peak of benefit. So this is what we're looking for in wildlife or livestock. We're looking for that optimal level to sustain health, maybe even improve health. Well, what's the problem with essential elements? Too much of a good thing. So you can overdo this and you end up in the toxic range. This phenomena here is called hormesis, okay? Something we need to appreciate for our essential elements. How many people have heard of hormesis before? Dr. Webb's nodding. He's not putting up his hand, but I figured Dr. Webb had, yeah. So, yep. So this is a really important concept. Biogeochemistry. So we've been talking about how do we assess the soil, the forage, the browse. Well, range, crop, and pasture management really require an assessment of these elements. So there's when you determine if the animals need supplements or not. Not because some guy in New Mexico increased his productivity and you're in Montana and you think, well, I'm going to give selenium supplements. You can't do that. It's based on the biogeochemistry. Please remember, some plants are really good at accumulating some elements, and that may not be a good thing because they can accumulate a lot and they'll overdose the animal, the herbivore. River drainages. For me in Alaska, it's all about river drainages. Geological sources to the herbivores, yes, terrestrial. These river drainages have their own geology, their own biogeochemistry, and in some cases they limit the essential elements. In other cases, it's overabundant. So for us, when we talk about these things, we're thinking river drainage is not pastures or ranges. Aquatic species also can be quite vulnerable to elements. So we study fish. I won't present that today, but there are some concerns about some of these dissolved elements, like after a forest fire, mining activities, they get into the drainage, excuse me, they get into the drainage and they cause problems for the fish. I'll show an example of chemical oceanography with the stellar sea lions. So the One Health thing is important. Here we're talking about the environment, animal, and human health are inextricably linked. I've accepted this, that we're not going to understand all the things that bring us together in health because it's impossible to disentangle or separate. So if you believe in this paradigm and you accept it, it really helps with your perspective of how the environment, animal, and human health are linked. So we call it One Health in our basic science and biomedical approaches. The Alaska Natives, they've been practicing this for thousands of years. They don't need a term or a phrase. It's their way of seeing the world. So now our team is doing animal and human health together, pregnant women in Mexico and their fish, subsistence food consumers in Alaska and their fish and their mammals, and they eat everything. It's wonderful. So they eat a lot of different species, and they eat tissues that would surprise you. Okay? They are very good at using a lot of animals and a lot of tissues from the animals. And also they're cool critters, that's the title, as environmental sentinels. Some of these are great at integrating the ecosystem and telling us what's going on. I need a drink. I need a drink. So hopefully you understand we're trying to do multiple things here at the same time. There we go. Thanks for adding that. All right, so this might be better. This is the Alaska perspective. This is from the One Health Research Institute at UAF. This ties in Alaska Natives with Alaska wildlife and environment. And in that center is the One Health. And so this is really important for us in Alaska because animals represent subsistence food, safety, sustainability, health of the ecosystems. I'm going to talk a little bit about conservation of the stellars and how we got tied into that. So I think the Venn diagram is better, but I had to go through the text. This is the marine perspective. So environmental drivers, the host populations, that's an orca, killer whale if you can't see it. And then environmental agents of disease, bacteria, viruses, chemicals, variety of things. Again, it's about the biotic and the abiotic interacting. Very important. So this is our perspective. I'll just put them all up there. So this is the Alaska perspective. The one with the dotted yellow lines is typically what people in low 48 think, oh, we eat fish. In Alaska, all that is consumed. So the pinnipeds are consumed, the fish are consumed, the invertebrates are consumed. So for One Health, it's the entire food web there we're representing. That's also representing how it's integrated. And that when we go to the top of the food web, to the piscivorous marine mammal, fish-consuming marine mammal, that helps integrate the ecosystem. And then that all affects humans. Humans and marine mammals consume the similar items. That's why we were funded by the commercial fishing industry. And then top predators aggregate numerous food webs. So really cool. So for the Aleutian Islands and the stellars, we're going to talk about Mercury, not the planet. What I told my kids was, you know, when you go to school, tell your friends that Dad studies Mercury, you know, like the planet. See, then I could be like a space scientist. Well, that didn't go very well. I wasn't. So anyway, selenium, and then the interaction between the two. We just did a recent publication on biogeochemistry of this. We study fish, cephalopods, crustaceans from this region as well. Not enough time to cover that. A very regal animal. Those males are amazing. And when they're chasing you, they're even more amazing. And then one colleague, when I showed this slide at College Station, he started laughing. Apparently, he owns a Fiat 500. Yeah, look how big that male is. Females are not small either. So, and that's what we would call a rookery. That's their typical home. And so, very remote area of the world. Here it is. Some people like to think this is a family. How many people know about pinniped biology? Yeah, they're not families, right? Those males form harems, and they will literally run over their young to get to another male that they don't want in their territory. So they form these harems, and that's kind of a PR picture, really. But this is their range, and some of their more permanent rookeries. They tend to be very rookery specific. They have high fidelity for where they were born, and that's helpful in us when we look at the geographical range of some of our issues. So a lot of our sampling's taken place in Russia, and most of us got out okay. Oh man, there's a story there. So this is the conservation side of things. We just didn't go out there because, oh, stellar sea lions are sentinels. Yay. No, we went out because for decades, 2 or 3 decades, the population is decreasing rather rapidly in this portion of the Aleutian Islands. Russia's over here somewhere. And here they were starting to stabilize or increase. So NOAA, the federal agency responsible for management of the stellar sea lions, was really trying to figure out what was going on. And so we keep updating these population estimates, the pupping trends, things like that. Things are not going well where the red arrows are. This is where you go, aw. Oh. Thank you. These are stellar sea lion pups, and when the adults go out to forage, or when the biologists show up, they leave, they aggregate like this. And so why am I mentioning this? Because this is a great sampling opportunity. We can sample many, many animals from one rookery at one event. If you dart a female chemical mobilization, just stop darting them, chemical mobilization of a male or a female adult, where does everybody go? In the water. That's it for today. You get that one animal. Do you see what I'm getting at? So we'll talk about that a little bit more. They're born with a natal pelage and whiskers. We're going to take advantage of that. But that's the slide. This is to verify they do come out with full pelage, full whiskers. And this is where I like to talk about pup power. They have developed whiskers and hair, and that allows us the statistical power I mentioned because we can sample a whole bunch in one event. It's the cohort of concern that we'll come to in a minute that the mom, the dam, she's transferring all of this to her fetus. It's in utero exposure is what we call that. And then, believe it or not, we can assess the dam's diet based on this, right? The fetus, which now is becoming a neonate, it's in between, is fully produced by mom. So we can assess her diet as well. So we learn a lot from the pups. They object a little bit, but they get over it. So we put them in these little troughs, and sometimes they're sedated, sometimes not. Some of them just are very cooperative. So this hair sample is where we also get our blood sample. So this is why we like to use the hair, for those 4 reasons there. So that's our collection site. It's probably not easy to tell where that is. I'll just, right there. It's very convenient for getting a blood sample, and we get lots of hair. Okay, here's the issue. This was a surprise when we started generating this data. This goes from east. I keep losing my pointer, sorry. But it's labeled up there above the screaming pinniped. There we are. west, and then the far east because it's Russia, right? Yeah, we're in interesting territory when we get to Russia. So you can see Rus is all the Russian sites. And so we go from east, which is southeast Alaska. I'll show you a map in a minute. And we go west, and then bang, the mercury levels shoot up in the hair, and then they come back down. We've published on what we consider toxicological thresholds for mercury, 20 and 30. Well, you can see we go well above that, and our median goes above that for this particular island. And so NOAA immediately said, we got a problem because this overlaps with the rookeries that aren't doing well. Like that. So if you remember the red arrows and stuff, they were all in this area. That's exactly where the high mercury is. So by biomonitoring sterile sea lion pups, it's a conservation question. It's an environmental contamination question. Is mercury one of the problems they're facing that their populations won't recover? So this is what we're working on, and we continue our efforts. The thing to remember about mercury is it bioaccumulates and biomagnifies. So bioaccumulation is more age dependent. Over time, the mercury increases in some of their tissues. Biomagnification, where do they feed trophically? Where in the food web are they feeding? It's more contaminants than mom. So with selenium, remember it's essential and toxic. In my business as a veterinary toxicologist, we're always seeing people make mistakes with selenium. They kill their herds. It's really unfortunate. There's dietary sources. It's unlike mercury, it's regulated through homeostatic mechanisms until you overdo it. And then things break down. There's a documented interaction with mercury. I didn't think this would be a crowd for biochemistry, but there's a whole groups of investigators looking at that. It's a defense mechanism against oxidative stress, and it has significant antioxidant properties as an element, but also with the enzymes it interacts with. This is a paper where we looked at a whole bunch of elements. It recently was published. This is the biogeochemistry I was mentioning. So we mentioned that mercury is very different by region. Well, turns out this pass is turning out to be very important. It's a pass in the ocean, so you're not going to drive through it. You're not going to see it. It's just a deep trench with altered currents in it. So when we compared these two populations, all the elements were different. This is like comparing a pasture in one region of Texas to a pasture in another region that has completely different geology, soil chemistry. Does everybody see the analogy there? So these groups of stellars are not just different by mercury. They're different by many, many elements. So now NOAA's asked, well, what do you think is going on nutritionally? How many nutritional studies have been done in stellar sea lions? If you said 0, you'd be really, really close, like right. Well, thanks for having hope. Yeah, so we don't know how to interpret this, but we're going to be working on it. Another reason I'm here is because we feel this is the same situation for some livestock animals, that we just don't have a real good grip on what it means for their health status. 11 of 12, I guess it's easier just to say, well, zinc was the same. Anyway, this blew us away. It was a cool paper. We did all this chemistry in our lab, which is going to be demolished in February. So much for good research. Oh, thank you. Another aw. You're good at this. So how do we interpret this? Well, we had to go to rehabilitation centers. A lot of these wildlife students are going, boo. Yeah, there's good and bad things about rehab centers. But with stranded harbor seal pups, we were able to closely monitor them, including their hair and mercury, blood mercury concentrations. We could finally look at severe and subtle outcomes. In this case, we wanted to look at neurological assessments, because in the stellar sea lions, we looked at oxidative status, immune status. That was all easy to do. Well, not easy, but you could get the samples to do it. There is a joke in here about doing neurological examinations of pinnipeds. They're not the brightest critters, right? So these are going to be things that are more innate. And I'm not going to go over that, but if you wanted me to come back and talk about it, we're publishing a paper on the neurological exam of these. San Francisco Bay, known for mercury contamination because of mercury and gold mining, all drained into San Francisco Bay. Which is interesting is we got the same dose response range. So this is very similar to the mercury concentrations we saw in the stellar sea lion pups. So I call it dose response. And these are the harbor seals from Central California. So we're fortunate we can do studies in both places. I would really like to have given a more thorough discussion on this, but I'm sure I'm running out of time. We found there was associations of mercury with neurological problems. Like I said, it's a pinniped. So we decided we would dive in a little deeper. So we developed a neurological exam specifically for harbor seal pups. We did a multi-year, more detailed assessment as a PhD dissertation chapter in a manuscript. Drum roll, please. Thank you. So, after all that, they had what we often associate with mercury toxicosis in people in lab animal models: decreased response to tactile stimulation, decreased movement, longer stay in rehabilitation. This study is now continuing, and we're going to try and figure out how directly associated is mercury with this. And guess what we're including to confuse things? All those trace elements we measured in stellar sea lions to see how different they are. That's the beauty of science, right? Answer one question, you generate 20 more. So on the moose, unless I should stop for pinniped questions or just keep rolling. Really? They're a captive audience, literally? You have guards? I'll get another drink, and you guys can think about if you have a pinniped question. And yes, they're cute, but they stink. They're all covered in fish goo and poo. Oh, we got one right there in the middle.
So, I'm not a specialist in wildlife at all. I just had a curious question. Whenever you saw that there's high mercury levels in your Steeler sea lions, did you by chance take, I don't know, a sample of the fish that they could potentially be consumed to see if it was a specific species and like what the cause might be of that?
Do I have enough time to give a detailed answer? Well, because I want to give a detailed answer is because, yes, we did. Yes, we took a look at their prey species and looked at mercury, but we've also done some other things. We've used stable isotopes of carbon and nitrogen to look at their feeding ecology and to sort out what trophic level they're feeding on. Some of the animals in the West are feeding at a higher trophic level. That's the biomagnification. So they may be getting exposed to higher level mercury because of the marine prey, right? Higher trophic level. Those fish also have more mercury in them too out there. That's one. The really cool study that we've done is we put units on these animals to specifically see where they're located and how deep they're diving. And we got mercury levels on them. And we're looking at their diving behavior and their foraging behavior related to their mercury exposure. Oh my gosh. Yes. Where they go and how deep they dive seems to be explaining their mercury exposure. I'm sure there's not a lot of marine ecologists in here, but it seems to be those animals that get to the benthos, the bottom, and feed on fish and maybe even invertebrates in the benthos, in the sediments, where we found that those fish that live in the benthos in some of these regions are higher in mercury. Isn't it cool? Isn't this just, I am such a lucky guy that I get to work with people that can put these instruments on animals and tell me where they're going, how deep they're diving, And all their behavior, it's amazing. Oh, and by the way, they instrument them in a way that they're collecting environmental data while they're doing all this, like temperature, so on. Anyway, this is fun stuff. We don't do that for the moose and caribou, by the way. It's more two-dimensional. Does that answer your question?
Yes, thank you.
We're sorting it out, and it's only costing you as taxpayer. Well, I better not tell you that part. Moose and caribou. So this is my large animal analogy for you. Like, I don't want to say they're a cow, but I think you know what I'm saying. They're ruminants, right? So let's just say they're ruminants, and they share some similarities. So for Alaska, we just got great news today about funding on caribou while I was sitting over there chatting with some faculty. We got more money to study caribou. And I have a feeling you guys would like to come up and help me sample. Yes? Yeah? You can tell by the hats and the shirts. Moose and caribou, we're very lucky to be doing these studies. And Texas, we would like to look at goats, horses, and cattle. We've been talking a lot about that today, that it might be time to take this approach and look at these critters in Texas. So we had discussions here about that already. Approved and updated reference ranges for the wildlife is very important because you're going to see some information here that leads to why this is important. The agencies need improved herd health and management. They need this information to know what the stresses are that are preventing a population from reaching their expected potential. I already mentioned 0 calf production for 3 years. Wildlife management guys, what's that mean for a moose population? Doomed, right? So we had to turn it around. And so that's really important. So we are proposing that this is needed, that we need better reference ranges for all ruminants and even some hindgut fermenters like the horse. So Texas and Alaska, I'm hoping to get some momentum on this. And, yeah, cool. Now we're talking about moose. So I like moose more than Steller sea lions by far. So this is our program in Alaska. It's run by ATRIC, the Aetna Intertribal Research Commission. This is Alaska Native based. And they're funding us to help them with understanding the health of moose. So we don't go out and shoot the moose ourselves. That would be problematic, like here in Texas, right? Oh, we're just going to go shoot a bunch of deer. No, we work with the hunters, and you'll see how this works. So we encourage the hunters to collect samples for us or invite us along to collect samples from their moose. So these stations are set up, and that's a picture of a hunter with one of these. You could even do that with producers here in Texas. So they're then going to collect the samples, fill out the data sheet. This will never work. Yeah, that's why we have 100 moose represented in the study already. The hunters buy into this. The hunting guides buy into this. They want to know about their moose. I think most people that are involved with hunters know they're highly ethical and care about the conservation of the populations they hunt. That's very true in this situation. One of my best examples is Ducks Unlimited. Talk about a group that invests in their ducks. Why did you get elbowed? Why did you get elbowed?
I interned more this summer.
Awesome. Aren't they a great organization? They are. We need Moose Unlimited. So my wife, when she saw this, called this badass chicks. I want you to know the women are like this in Alaska. Okay? They are rugged. They are rural. They're the ones who are pushing this project. It's amazing. So that's what's a moose that's going to end up in someone's freezer. So they are excellent. And so it's not about male hunters doing all this. It's about males and females working together as families. And it's really cool to watch. Isn't that beautiful country? Even looks better with meat in front of it. All right, moose samples. They're very dedicated. So they get a chunk of hair. We talked about hair with the Stellars. Muscle, well, that's important because that's what people eat. That's that food security thing. Kidney. Elders love kidney. And kidney is really important because it's a filter, right? Filtering organ. So that's going to tell us a lot about what elements they may be exposed to. Liver, there's nothing better than liver for looking at element status, to be honest. And people eat it too. What? Why do we have the incisors up there?
Tooth health is a good indicator of bone health.
Yeah, okay. Yeah, what else? We cut them, we cross-section them, and we age them. Good. Yeah, so and then we collect blood on these blood filter paper things that revolutionized how we do our research. So we'll come back to a picture of the teeth. So then we get these packages, and for us, we look at the mineral, sorry, I said mineral, I mean essential and non-essential elements. And there it is. So there's the lamina, right there. So that's how many years old this critter was. We'll call it an age estimate, but it's pretty good. It's a pretty good way to estimate cervids. So the concerns of the tribal group is animal health and food security. The reason we're looking at age is some of these elements bioaccumulate. So we need to correct for age. Some of these moose can live a long time. So these are traditional ways of hunting and processing moose. So you have an appreciation of what it takes to butcher these guys in the field, and you got to pack that meat out. So some of these guys use military operations. Others are going to sled dog in, well, when there's snow. Sled dog in or hike in. A couple of my friends, they paddle in kayaks. This is a lot of work to haul a moose out. Some use high technology, and so here's another one of our moose stations, and then collecting the jaw or the incisors. As sentinels, we want to look at cadmium and copper. So again, these environmental sentinels. Cadmium is high in the kidney, and again, that's why we age them, and in part is to look at age-related accumulation. So we are concerned, I used a fancy word, sorry. We're worried about kidney function and food security when it comes to cadmium in the kidney. These are animal health and human health issues. Does location matter? That's why we need to age normalize the data to see if there's a cadmium source out there that we're not familiar with. This was troubling, marginal deficient hair, liver, and kidney as a preliminary assessment. So again, you need to look at location, age, time of year. This would be true of cattle or goats in Texas, that you may have copper deficiency based on pasture or range conditions. In our scenarios, it's based on river drainages, based on studies I did in moose and caribou before. It really seems like river drainages are driving these differences in exposure. So again, it comes back to biogeochemistry. Soil science. Large animals as food? Well, this is probably not very popular in Texas either. They hate it in Alaska. So when it comes to food, and we need to look at dietary practices. Well, unfortunately, it starts with the ammunition used. Why? What?
Like, are you talking about like basically like ammunition, like it's going to just kind of obliterate the organs?
Yes, that's one concern we have is tissues not being sampled where there's been trauma. But what?
Bullets are made out of lead usually.
Yeah. So it's that
And copper jackets.
and copper. Okay, yeah, you're nailing all of it. So there's been studies done in Scandinavia to show that this is significant. So if someone's worried about a lead mine and we go shoot all the caribou with lead bullets, what am I going to be able to tell them? Yeah, you shot them with lead bullets. But we can use lead isotopes maybe. But lead is also not good for people to be exposed to. In Alaska, this is very unpopular to bring this up. I probably need security. But some people are switching to copper bullets because they've figured out that they're introducing lead into their meat. Okay? Remember I said liver and kidney is consumed in Alaska, along with other things? Raw tissues are what we're assessing now, but eventually we're going to look at food processing. Okay? So we're going to eventually look at how they process foods into the fork. That's what's important to the human consumer, not the kidney as it comes out of the moose. Store-bought foods. A lot of people in Alaska think store-bought foods are contaminant-free. I've already done one study that said, thanks for playing. No, they are not. So where they're harvesting these moose, they've decided they're going to go to the store and they're going to tell the elders, you no longer have access to your moose and caribou. What would you buy to replace those subsistence foods? They're then going to send them to me to analyze. Isn't that clever? We did that in Barrow, Utqiagvik, which is as far north as you can go in Alaska. So we're going to show them that these alternatives are not really any better. And so that's really important in giving this public health message. Woo! I did it. Did I do it on time? All right. I did good. Maybe we should go back to the badass chicks. They are wonderful people, too. They're not... If there's a hand up, there they are. That's Emily and Tamara. Yeah.
So do you have to control for the sex of the moose, or is it always a male?
Okay, so thank you. For the pinnipeds, it's been shown that there are sex-related variations that may be playing out. For the Arctic ungulates, we have not seen that. But for the pinnipeds, there is something physiologically that may be going on. Those pinniped females, wow, they're real moms. They get real fat, they have a baby, they get real skinny. And then they have to go out and forage while they're nursing. So these moms go through serious tissue changes that the males don't. So that might explain some of what we're seeing. They dump a lot of stuff into that neonate through their milk. Oh, I see one back there.
Yes.
Hi.
Hello. I have questions when you're measuring, so you focus a lot on the females, but are you, especially when they're dissecting or cutting or processing the moose, you can take a couple testicular samples, and because isn't there mercury, testicular, it'll go to the testes. I think I read a paper that it can lower semen quality.
So we would probably have to get our preliminary data here to justify what would be the next samples to collect. And reproductive organs, the tribal management group and Alaska Department of Fish and Game would probably want that. While I was visiting Glen Allen 2 weeks ago, I'm sweating, by the way, because I acclimated to Alaska. They decided that even though it may not be culturally appropriate, we might start sampling the fetuses whole so that I can necropsy them. Yep. It's not culturally acceptable, so they might let a white scientist get away with it. To be honest, but if someone in their community did that, that would probably cause a stir. They're considered, I don't want to say sacred, but they're treated with respect. But they might let me get away with it. Yes?
This is more about the Steller sea lion. So have you done samples for their predators as well, like killer whale? I know I know sharks are a big
Yeah.
predator. Do they have higher mercury levels as well?
That's an awesome political question. So, why are the Steller sea lion populations going down? Well, Senator Ted Stevens, who's no longer with us, when he found out that killer whales and sleeper sharks preyed on Steller sea lions, He went, see, it's not the fishing industry. So yeah, politically, the idea of those predators impacting the stellars has come up. And there is documentation of sleeper sharks, orcas, but there's also bycatch. Does everybody know what bycatch is? When people fish, they inadvertently catch something. There's also some sterile sea lion bycatch. So yeah, there's been investigations into that.
What I was asking was their mercury content? Like,
Oh, sleeper sharks are high.
sharks are fairly resistant. They have a high mercury content in general.
Yeah, in general, yeah. So yes, their mercury concentrations are high, and the orca skin biopsies have been high. We can't, well, when they're stranded and they die, we can sample them. But normally we get biopsies. Yes, sir.
I'm from engineering.
Mm-hmm.
Do you have you heard some good method to remove mercury from feed? If, for example, like you said, San Francisco Bay area.
Yeah, getting mercury out of the environment is something a lot of people are exploring, and they want to tie it up with other minerals. That would be the non-organic forms of mercury. When it comes to the mercury we're talking about in the fish food web here, it's called monomethyl mercury. It has a methyl group on it. That's what makes it bioavailable, is it hops on an amino acid, and it mimics another amino acid, and we carry it into our systems. The monomethyl mercury one is more difficult to consider how we would tie it up and get it out of the system. But the inorganic forms we can. And there's people working on that. Yeah, so there are places where this is going to be called for. Things like sulfur are contemplated. You want to tie it up, have it precipitate out and collect it. You could use gold. Anybody want to use gold to get rid of mercury? You know that mercury was used to harvest gold. That's why I'm saying that you could do the reverse. It'd be really expensive. Yes?
So you said that, that getting your information for the kiblets or the baby sea lions or whatever,
Pups? We have babies, they have pups.
Okay, great. For the pups, is a lot similar to getting it straight from the mother. For the moose, the 130 ounce limit, do you think it would be, efficient to get it from their calves? Or
Yes.
is it not practical? Is it how?
There's many reasons we want to get hold of the fetus. And one is, if they're copper deficient, is the fetus copper deficient, and is it going to suffer from problems? In most ungulates, mom gives a lot of copper to the fetus for her growth and development, for their growth and development as a calf. So the calf should come out copper enriched. If it's not, it's already at a disadvantage. And in Alaska, you are not given much opportunity to be deficient. So that would be the copper. The other question is this cadmium transplacental? Can the cadmium get into the calf and cause problems? Well, the evidence so far is maybe not. But we haven't done many fetal exams to determine that. And also, if the copper is deficient, is there something associated with the fetus that would tell us that it's compromised? So that's why we'd want to do a complete professional necropsy to see, is this fetus at this time of year at the expected parameters we would expect. Sometimes calves are born half their size they should be. That's bear bait. Yeah, they're going to have a hard time. They won't make up that, these guys know this, they won't make up that difference. They can't. It's impossible. Yes?
So with that excessive cadmium and then the deficient copper, did you see an increased amount of abortions or neonatal deaths?
We did in the Colville River. We went in, not in the Colville River, the moose associated with the Colville River. It's not good to go in the Colville River in the middle of winter. What we saw was that This was really a fun project, even though it was tragic. Okay, we went in and we did pregnancy examinations looking at blood levels of certain hormones. The moose cows were pregnant. That was the first question, right? Because if they're not having calves, well, if they're not pregnant, duh. So we went in and said, okay, they're pregnant. And then we monitored them having calves and how long the calves lasted. And they were gone very quickly. So either they were doomed to mortality or they were very vulnerable to predators. But the bottom line is it was zero recruitment. After we did this study, the copper levels started to go back up, and they started to have some calves that survived. Does that help answer your question? Yeah, it was. I'd only been in Alaska 2 years, and here I am flying around, darting moose, pregnancy checking them. Man, being a veterinarian in wildlife world, it's like you're always dealing with tragedy and you're having a great time. But that's how the career goes. Really appreciate all your attention and the attendance and the questions. I'm not sure, are we done?
Any other questions?
I do have a dinner meeting to attend. I understand it's going to be very formal. Thanks, everybody.